Students, welcome to my YouTube channel. So today our topic is gene interaction. We'll be discussing about the gene interactions. There are two types of gene interaction. One which occurs between the alleles of one gene. It is occurring between the alleles of one gene. That is why it is also known as intrageneric interaction or interallelic interaction. Another is your intergeneric interaction. It occurs between the genes. This type of interaction occurs between the genes. As a result, it is non-allelic gene interaction. They are not alleles, then also interaction is going on. So we discuss it as non-allelic gene interaction. So this is a mind map which I have prepared for you to make you learning easy. So today in our topic, we'll be just understanding what types of the interactions are there and what are the categories present under them. Now see, in the intra-allelic gene interaction, one is your pleiotropy. In the case of pleiotropy, one gene control many characters. This is pleiotropy. Another is multiple allele. Multiple allele means one gene has got more than two alleles. So here one gene has more than two alleles. Another is your dominant interactions. If you remember, Mendel has discovered only one type of dominant interaction, that is dominant and recessive relationship. According to the Mendel, in F1 generation, it resembles in only with one parents. So according to the Mendel, F1 generation heterozygous resemble only one parent, that is dominant recessive. If F1 does not resemble any parent, then we call it as incomplete dominance, which is discovered after the Mendel work is discovered. Another is your codominance, in which F1 resemble both the parents. So these two interactions are known after the Mendel work is rediscovered. So these two interactions are known after Mendel work. Another is lethal gene. Now another is your intergeneric, means between the genes, or with non-allelic gene interaction. One is your complementary gene interaction. So today I will just focus with this only, with the examples and the ratios which is modified from the Mendelian dihybrid ratios. So you can say here it is actually two gene interaction. This is two gene interaction where complementary ratio is there, which is studied in the flower color in sweet pea, and the ratio is modified from the Mendelian 9:3:3:4 to 9:7. Another is a supplementary gene interaction, which is studied in the seed coat color in bean, where the ratio is modified to 9:3:4. Now recessive epistase. Epistase is actually non-allelic pair. Suppress the expression of another non-allele. Then such type of interactions are known as epistase. Here it is working with the different alleles, means it is working between the two genes and as a result between non-allelics. So it can be recessive in nature, it can be dominant also in nature. So recessive example is coat color in mice, 9:3:4, and dominant is fruit color in summer squash, whose ratio is 12:3:1. Detail all will be discussing in the next videos. Next is a duplicate genes. Duplicate genes are the genes which is studied in the capsule shape in shepherd's purse, and the ratio of the Mendelian was modified from 9:3:4 to 15:1. Inhibitory gene interaction is studied in leaf pigmentation in rice, and collaborative gene action is studied in the comb shape in poultry. So all these interactions are two gene interactions in which dihybrid ratio is modified. The dihybrid phenotype ratio is modified. I hope this mind map will help you to take a quick revision of the gene interactions, which are of two types, intrageneric and intergeneric. Just remember, intra means it is occurring in one gene only, and intergeneric means between the genes, means two genes. So it is occurring in two genes. You can say two gene interaction. In the next video, we'll be discussing the intrageneric interaction in detail with examples.